Fossil record. So we can look at changes in species over time. We can see one species go extinct and another species replace it in the fossil record. There is a fossil species that you got to take a look at yesterday, a species that exists only in fossils. What is it? Yeah, well, you know, it's the best I could do fast. So anyway, T-Rex, it's a species only known in fossils. There are no living T-Rexes. Sadly, it'd be really cool to see one from a distance, from a safe distance, enclosed in something T-Rex proof. Only known from fossils. But yet, we can trace the evolution of dinosaurs. And when we go to the Carnegie again, we'll look at the evolution of dinosaur species, and we can see what species just disappeared, like our old friend here. Fossil record, what's the second line of evidence? Structural evidence. And for structural evidence, we have, and I think somebody's doing the Vanna White back there that I was going to do. Is that what you're doing, Peyton? Look at the structural evidence. Oh, I thought you were giving a clue. Look at your fine limb there. What a nice limb you got. That limb that looks a whole lot like a cat's limb or a bat's limb. What kind of evidence are we talking about? Homologous structures. Same shape. Same shape in those structures. The other kind, anybody got goosebumps? Vestigial structures. So we've got fossil evidence, structural evidence, and the third line of inquiry that we're following here, chemical evidence. So that's where we left off, and the first kind of chemical evidence we talked about was, in fact, our old friend DNA. So when you did Lizard Lab 2, the second module, remember what kind of tree you built? It built for you, actually. You didn't have to do it yourself. Phylogenetic tree, that thing. That thing! What was that phylogenetic tree based on? They were based on DNA. Yeah, the phylogenetic trees were constructed based on similarities in DNA. So it's not just that they use the same DNA, that all living things on Earth use the same chemical, but that we can look at similarities in DNA, and based on that, we start to get a sense of how closely related species are. And on those phylogenetic trees, the things that branch off at points that are closer to one another share more DNA. And we'll talk a little bit more about that as we go. Okay, so you've all seen cladograms. You've all worked with cladograms now. How many of you felt like you pretty much knew how to read a cladogram the minute you saw it? It was very obvious to you. Okay. I'd say 25 to 40% of students say, like, okay, yeah, that was just so intuitive, I didn't need to have it explained, it just made perfect sense. For everybody for whom it wasn't an immediate, like, oh, that makes so much sense. One of the things I like about that tutorial is if you get it wrong, what does it do? It says, oops, try again. Oops, try again. And it guides you to the right answer. So how many of you felt like you gained some skill at reading cladograms using that, through its use of, whoops, no, whoops, no? Did that help you? Remember I said that cladograms are important. I mean, they're important in biology to understand evolutionary relationships, to arrange the information we have about shared structures, about homologous structures, and about shared DNA sequences. The state of Ohio also thinks they're important. There were about 10 cladogram questions on the air test last year. There were only about 30 questions on the test. So about a third of the questions had a cladogram. Another third of them easily had DNA sequences and protein synthesis. So you know how we did that with bacteria and protein synthesis? We're going to do it again because I want to make sure you're ready, and I want to make sure you score high enough that you don't have to retake and that you get your ticket punched on the first shot. That's my goal. But anyway, cladograms are a way to show that relatedness, and they provide evidence of shared DNA. Very often cladograms are also done on structures like the one that you saw. The closer two things are on a cladogram, the more DNA they tend to share, and the more structures they tend to share, and you saw that on yesterday's activity. So the thing to remember is that distance on the cladogram is more about distance back to a common ancestor and less about being side by side. So that's just a thing to remember. Okay, one last point. More chemical evidence. I'll read it, then shut up. Hemoglobin, the chemical that moves oxygen in blood, is used in all vertebrates and a few invertebrates. And human and gorilla hemoglobin is 1 amino acid different. It is nearly identical. Has anyone here ever heard the term hemoglobin before? You might have had your hemoglobin tested if you had blood work done. Hemoglobin is essentially the wheelbarrow that carries oxygen to every cell in your body. So if your hemoglobin is low, you get tired very easily, your muscles don't work correctly, because your cells aren't getting the oxygen that they need to function. And the chemical hemoglobin is used in everything that we know of that has a backbone. We've never found a species that's a vertebrate that has a backbone that doesn't use hemoglobin. There are even a few invertebrate species that use hemoglobin. So right there, that's evidence of common ancestry. Humans and gorillas, who are more closely related than a lot of other vertebrates, they're both primates. This is an actual image of hemoglobin. It's a folded protein, and if you remember back to protein synthesis, you have a bunch of different amino acids that line up in particular ways and then fold, and it looks like somebody took some yarn and some old telephone cord, tied them all together, but that very specific shape is critical because if it's not shaped exactly right, you can't transport oxygen in your blood. And there are diseases where people mismanufacture hemoglobin, and without correction, I mean, if they mismanufacture all hemoglobin, they die. Period. That's fatal invariably. There are some diseases where like a percentage of the hemoglobin produced by someone's body is malformed, and then they just sort of battle this ongoing issue. Human and gorilla hemoglobin are almost identical. There's 1 amino acid of difference. How many DNA sequences are represented by 1 amino acid? Vote with your fingers. Remember protein synthesis? A, U, G, 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 U. 1 amino acid is coded for by 3 little DNA bases. So it's 3 bases different in the cells in gorillas that codes for amino acid compared to ours. Okay. That's actually where we finish the notes. After this test, what we'll start on is natural selection, which will seem eerily familiar to you, I'm quite sure. That's where we're going to stop.